So my name is Shannon Mumenthaler. I'm an assistant professor at USC. So I was trained as a cancer cell biologist, but I have to say that about 5 years ago I interacted with physical scientists through the NCI Physical Science and Oncology Center, and I actually evolved as a biologist as well. So I study cancer as an evolutionary process, and as a biologist I've evolved into more of a quantitative biologist. And so I'll get into kind of the experimental techniques that I use, but really my focus is on a lot of the work that I'm interested in is this major clinical problem in cancer, which is drug resistance. And this drug resistance really stems from tumor heterogeneity, both at the genetic level, so there's individual tumor subclones, but then there's also the heterogeneity of the tumor microenvironment and how those intersect. So how does the microenvironment actually lead to certain subclones actually evolving and taking over a particular tumor? So in this case, you can see in the top figure, we start out with admixed populations of cells, some that are resistant to a therapy, some that are sensitive to a therapy. We apply individual selective pressures, not only looking at drug, but also looking at gradients of oxygen and nutrients and other stromal cell types, and how all of these influence the fitness advantage of individual subpopulations, leading to resistance. And so the quantitative platform that I'm using is a high-content screening platform. So we look at thousands, hundreds of thousands of cells. We can do this in a high-throughput fashion, so we have 96 well plates. We can, in each of those wells, we can have a different perturbation, so we can look at how oxygen alone, as well as oxygen in combination with drug and other environmental perturbations, influence the live and dead cell counts, and we use this data to then estimate birth and death rates. And I collaborate with Jasmine Fu, who has a stochastic branching process model where this data feeds into that model, and we look at designing optimal treatment strategies. I think that's the ultimate goal. Yeah. Oh, well, no, I mean, what's the question?
I'm forced to try to answer. So
yes.
You're doing some modeling work already. Are you openly looking for people to collaborate with to extend to different types of spatial modeling work?
Absolutely. Well, I also, so Paul and Farzan already mentioned some of the other projects that I work on, so I do collaborate with other modelers as well. So yes, I am all about working with physical scientists. I think that's where we need to go in terms of oncology. So. Yes. And actually all of my work now, I very rarely do any of the traditional cell biology assays anymore. A lot of it has changed because of my interactions with quantitative quantitative scientists. I think they've changed the way that I view biology. We need to be looking at it in dynamic, quantitative aspects. And I think as my training, it was more qualitative and static time points. So I think it's important. Yeah.
Are we starting? Okay. My name's Jeff Clark, and I work in Peter Zanstra's Stem Cell Bioengineering Lab at the University of Toronto, and that was sort of a great introduction to my stuff, Shannon, thanks. I'm going to do this off the cuff to try to focus it a little bit more on the way we generate data and then what we try to do with it. But we're an engineering lab, so we're interested in creating products, and those products are lots of stem cells and lots of differentiated progeny of those cells. And that's really for regenerative medicine, where we can use those products as sources for drug discovery, for tissue regeneration, and more recently we're using it to look at developmental biology as well. So one of the big issues in stem cell culture is that the colonies that we get are very heterogeneous. So here, this is just a chloropleth showing the expression levels of a particular gene that's involved in pluripotency. And you can see that really the cells are all over the place. And so we don't know what that does with respect to how these cells differentiate and how they maintain their pluripotent state. So what we do is try to develop models of the intracellular genetic regulatory networks that control pluripotency, and then link them to the extracellular variation that's known to exist within the microenvironment of all the cells. So on the data aspect, we also develop new technologies for patterning cells in specific spatial orientations. And we can do this in a high throughput manner using high throughput content screening. So we also use 96 well plates, but we pattern about 16 individual colonies in each of those wells. So we have huge numbers of colonies, and we spatially separate them so that they don't interact anymore, and we don't have to worry about that issue. So we can engineer these pretty much however we want. And we're also working on assay systems for 48-hour assays where we can track their cell fate in essentially real time. And we're also working with collaborators that are allowing us to track 3D colonies of cells with live cell tracking as well. So
very
Oh,
cool
yes.
stuff. When you actually go into to collect this data and the different types of tracking, what method do you use to share that? Is there a specific format or standard that you use to share that, or is it sort of just the picture? Or
we are starting to develop our own code to actually do the image analysis on these things, and that will all be open source. But generally, the equipment that we use is off the shelf. We just buy it from whatever biotech company that produces it, and they have their own data formats. Unfortunately, that's the way it sort of works in experimental stuff. You don't have, every company uses different formats, and they don't like to talk to each other at all.
Yeah.
So that is a real issue for us. But luckily, there are only a couple different types of equipment that do this, so we don't have a massive, massive problem with that. So.
Hi everyone, I'm Babak Momeni, and my general interest is in microbial communities and how species interactions such as metabolite exchange in these communities can lead to community properties such as spatial organization of the community or their general biochemical activities. And what we have done before, as Wen-Ying talked about it in more details, is using an experimental system that was based on engineered yeast strains with defined interactions, as well as the corresponding simulation method, which was a simple agent-based model that included processes such as nutrient diffusion, consumption, and release, as well as cell activities such as cell growth and death, as well as cell rearrangement, to look at the spatial organization of cells depending on different types of interactions in these communities. And what we found was briefly two main trends. One was that interactions between different species can lead to distinct types of patterns in the communities. For example, competition creates segregated patterns. Commensalism created layered patterns, and cooperation leads to intermixing of the two species in the community. And we also found that in a simple model of cooperation and cheating, these patterns actually have functional consequences. They can help cooperators get away from cheaters. And moving forward, I'm interested in looking experimental and simulation models to look into other community properties, for example, look into species coexistence in communities or invasion of communities by other species or by variants or mutants in the original community. And for that, my personal interest is to develop simple models to look into those properties. But I'm also interested in collaborating with others and use many cell modeling as a point of reference to compare predictions. That's it. Thanks.
So we've heard about how there was a specific question in mind where simulation helped you. Is there any point in which running a simulation then fed back into specifically what kind of data you were going to collect next?
Well, practically doing the simulations was a lot helpful in designing the experiments because we knew what like cell densities were likely to give us the results that we wanted. So it helped a lot to just mainstream that part of the process.
So my name is Stefan Hömer, and my group at the University of Leipzig in Germany mainly focuses on developing multi-scale liver models that are tightly parameterized by experimental data. So to give you a quick overview of what we do is we usually integrate at the molecular scale pathway models given in SBML. We integrate them in each of our individual cells. So we have different cell models starting from the spherical cells to more complex cells with triangulated surfaces here. On this cell scale, we focus on cell proliferation, cell death. We have the cell-cell interactions, of course, and then this all integrates into the tissue scale, where we have certain architectures that are parameterized by experimental images like CT or confocal data. And we add at this stage also the blood flow. This basically is a 1 millimeter edge length cube of liver tissue. And then if we want to use this tissue to actually run a simulation, we have to add the body context because blood leaves the liver, recirculates through the body and re-enters it. So we have additional models on body scale. So Our main challenges is the model selection, so what is important for specific biological problems, how to define the interfaces between all those submodels consistently, how to parameterize these with terabytes of 2D and 3D image data, and of course the usual code and implementation complexity. So unfortunately this should be a video showing you the regeneration of a liver lobule, but yeah, so that's only an image, so if you're interested, please join me and I will show you the running video. Thank you.
In this case, you're the only person I knew that before I picked the name. So one of the things I wanted to ask was, you're using the different images, for example, maybe for instantiating a model. When you have these different images, what kind of scale, like how much of the liver can you see in one image, and then what's the resolution? Like, do you get down to single cells, or how?
Yeah, it rather depends on the technique that you use. For example, in bright field images, you can see the whole liver. So, for example, the whole mouse liver, but only in 2D, down to the resolution of a single cell. In confocal, for example, you have 3D information, but you only see certain aspects, like the cell nucleus, only to a depth of 200 micrometers, for example. So you really need to combine different modalities of image data to get the whole picture.
People might want lunch though.
Okay. I take times. Okay. One of my project is the breast cancer carcinogenesis. The breast cancer is a multi-year, multi-pass disease process through the normal stage to pre-malignant stage into malignant stage. So this involves accumulation of genetic mutation, epigenetic alteration, and aberrant cell signaling pathway accumulation drives cell from this stage to this stage. So this kind of process takes multi-years, maybe 10, 20 years. When I try to apply my technology to understand breast cells 15 years ago, I look at those mainstream studies, I feel like it's not very practical, because the mainstream use a high dose of carcinogen to treat cells. Those level at a micromolar to millimolar level. That means if we smoke, we need to have 10,000 cigarettes a day in order to cause the cancer. So it's not practical, because our body only accumulates picomolar to nanomolar level of carcinogen in our bloodstream or urine. Therefore, I established another model, use breast epithelial cells as a template, and then treat cell with carcinogen. Those carcinogen at the picomolar or nanomolar level. And treat them in 24 hours, and then we wash away, make cell to proliferate, recover from the treatment. Through this kind of exposure, 5, 10, 15, 20, 30 cycles. And then we study what changed in those cells. Induced by carcinogen in single exposure, we study transient change. And after those accumulated exposure, we study permanently acquired cancer properties. All these properties we establish as endpoint can be measured. We can measure this point, and then to determine the progress of cell from normal to pre-malignant to malignant stage. So this endpoint, we use more than 10 endpoints. Okay, every point is one of these cancer-associated properties. So through this part, we identify those endpoints, same time, and then we study what preventive agent can block this process. So come out the true prevention concept. Instead of you have a tumor cell, and then the prevention means prevent the small number of tumor cells become big tumor cell. In the conventional prevention. And our prevention is prevent cell from being carcinogen, exposed changes. So we identify those dietary reagents which can block this process, block the transient endpoint, and then block constitutive carcinogen endpoints through this whole pathway. Once the cell still enter into malignant stage, and they can form tumor in new mice. And then we identify selective agent to kill tumor cell because one cell become this stage is far different from the normal cells. Therefore, we utilize the differences to kill these cells. Currently, we use specific agent instead of suppression this upregulated pathway or mutations. We try to enhance the mutated part through the reactive oxygen species. Because RS at the low level is mitogenic. Higher level become mutagenesis. The high level is lethal. So in the tumor cells, the RS level is already elevated. So we use drug to enhance RS, to pass the lethal level to kill transformed cell bypassing normal cells. And then we have used the combination to do the job.